In order for us to understand why we should be performing a protein assay, it's probably a good idea to first understand what a protein assay is. And to understand what a protein assay is, it probably makes sense to first understand what an assay is. An assay is an estimation of value based on the analysis of something for its specific components. When we run a protein assay, we are therefore estimating the protein concentration of a sample. When we think about the different assays that we run in the lab, a lot of them have the goal of estimating how much protein we have. Western blot, protein assays, protein arrays, INSA Westerns, and so on. So in theory, maybe all of them could be called a protein assay. However, for most people, when we say protein assay, it usually means estimating how much protein we have on a lysate. And when it comes to the Lambda U courses, that is exactly what we're going to do. So whenever we say protein assay, we're referring to estimating how much protein we have on a lysate. Now let's keep in mind that there's a lot of different protein assay options out there. So we want to make sure that you choose the best option to suit your samples. So first, let's understand why should I really perform a protein assay before every Western blot? Well, because if I don't, there's a very good chance that I may end up overloading my gel, which means I may get poor stacking, poor separation, and overall just a blot that doesn't look that great. There's also a good chance that I may end up loading either too much or too little protein. That could lead to severe transfer issues. Another potential issue that you could have from loading either too little or too much protein is that you may fall outside the linear range of your Western blot. Being within the linear range is very important because it's what guarantees that there is a linearity between how much signal you see on your membrane and how much protein is actually there. Running a protein assay may also help you notice in case you had problems with your extraction protocol, because it may help you identify when you have contamination with DNA, for example. Now, even if you're not planning on running a quantitative Western blot and only a qualitative Western blot, performing a protein assay is always going to ensure that you have an even load, run, and transfer, and that you're never overloading your gel or having transfer issues or being outside the linear range. So never forget that normalization against an internal loading control does not eliminate the need to know how much protein you're loading on each well of your Western blot, as normalization can only account for small pipetting errors and transfer variations. So even though every protein assay has an intrinsic variability to it, whatever error may happen, it will happen evenly throughout all of the samples. Ultimately, that means that you will be loading the same amount of protein on every single well. Also, using other methods different than a protein assay to try and account for how much protein you're loading is probably not a good idea. For example, if I try to use the number of cells that I have on a dish as a normalization method for how much protein I'm loading, I'm not going to be taking into account any variability that I may have on my extraction protocol or even while I'm storing my samples. Again, even though running a protein assay is going to take you a little bit of time and a little bit of extra effort and resources, ultimately it's going to guarantee that you're getting accurate, significant data and ultimately saving time, samples, and resources.